Hello, my name is Emma White, and today I'm going to be teaching you about gene therapy, or more specifically, how do genes work and how does gene therapy work and how do scientists use it in order to create complicated illnesses such as LCA in patients. Now, before understanding the complexities behind gene therapy, it's first important to understand what DNA is and what it does. So, DNA is best described as our body's personal instruction manual and tells everything what to do. DNA is packaged and organized into chromosomes, and a part of the DNA and the chromosomes instruction manual is to create proteins. And these proteins basically tell your body everything they need to do to function, and they all have their own individual job. So the proteins may tell your organs what to do, or they can even control your physical appearance. So, although it would be really nice, the body is not perfect. And sometimes there can be problems when the DNA creates proteins and the instructions get a little messed up. These problems are called mutations, and a mutation occurs when parts of the DNA, such as the proteins, might be missing or when they add a complete new part. Either way, it causes the cells to no longer function properly. This can lead to physical abnormalities or genetic diseases. It can cause webbed fingers and toes or even cancer. Now, I'm sure your main question is probably, can mutations be treated? And the answer is yes, in some cases. In order to treat genetic diseases, you must undergo gene therapy, which is a treatment method that aims to cure a disease by targeting a person's faulty genes. This is typically done by inserting a virus to carry healthy genetic material to your faulty genes. The hope is that the gene therapy will restore normal function by either replacing what was missing or removing what was extra. To put it plainly, gene therapy inserts the correct genes into our bodies. And don't worry about the virus, they deactivate the part that makes you sick and they just use it as a host to carry the material. Now, a main example of gene therapy can be seen in the disease Leber's congenital amaurosis, otherwise known as LCA. And this is a genetic disease caused by a mutation on one specific gene. And this disease causes the body to have the inability to produce the necessary proteins that are needed for our eyes to function. And because of this, LCA not only causes vision loss, but eventual blindness. Now, gene therapy was kind of put on the map as a treatment for LCA by one young girl, Molly Troxell. Molly was diagnosed with LCA when she was very, very young. However, her parents simply did not want to accept that blindness was her fate. Molly was put into a trial treatment that involved inserting a virus with the correct genetic material directly into her eye and into her retina. And the hope was that it would cause the protein to gain function and bring her sight back. And thankfully, this did work, and Molly was able to regain her vision. This popularized the concept of gene therapy in medicine as a whole. Now, although gene therapy sounds perfect and great in theory, there are many people who question it, and there are a lot of common misconceptions. One main one being gene therapy is just used to create designer babies, which are when scientists try to change the genes by adding or removing proteins of unborn babies during IVF. This is simply incorrect. Scientists are more focused on researching important gene therapy topics, such as curing diseases, rather than creating superhumans. Overall, gene therapy is very important because not only does it save lives, but it improves the quality of life. It treats the problem at a direct source. It encourages scientists to search for cures and have further conversations about diseases. Overall, gene therapy may seem very complex, but ultimately it's just as simple as going back and correcting a typo in an essay. It's a seemingly simple fix. Without gene therapy, we wouldn't have treatment for such horrible diseases like cancer and LCA. Thank you, and here are my sources.